Thanks for indulging the icebreaker, and I hope that you had fun talking with someone who hopefully you didn't already know well. And now we first have the working group from Molly, Albecker, and the rest of the individuals who are listed on the Zoom slide there, who studied selection across the life cycle. And so Molly, I turn it over to you now.
Hello, everyone. Can you hear me? All right, Katie? Great. So I'm really excited to talk to you from Utah today. I wish I could be with you, but the gods of moving to Texas, that was not to be. So I will be presenting the conclusions and the findings that we came out with in a paper that arose during the 2019 RCN workshop. And I'll be presenting, but I really want to highlight the impact and the influence of all these people listed. Letitia was a co-lead with me on this effort, and then Stacy, Sam, Matt, Holly, Mary, Katie, and Adam were just really pulled more than their weight in getting this paper rolling and creating these ideas. So keep that in mind, please, as we move forward. One second, let me advance the slide. There it is. So the origin story, it started there at Shoals. I actually wasn't there. I had just given birth to my now 3-year-old. So as people were coming up with their ideas for things they wanted to work on, it became clear that a lot of people there worked on animals or algae or microbial communities that had this complex life cycle. And instead of focusing on the specific details of the life cycle, the question that sort of emerged was, well, how does having a complex life cycle, period, affect your ability to adapt to a new environment? But as you can see, and this is a sample of some of the taxa that the people in our group worked with, there's a lot of diversity in these taxa. And so one of the first questions that bubbled up, and the main question that bubbled up, was how does having this complex life cycle affect your ability to adapt to a new environment? But it very quickly kind of became this dual question of, well, how do we study it and how do we talk about it, given this diversity that we see across researchers and across interests and across systems, et cetera. I won't belabor that point because I know that there's a panel later on today that will be talking about the importance of language in unifying study. But that clearly was an issue that needed to be overcome to study this question. So what we ended up coming up with was a conceptual framework, a way to think about the ways that selection could be acting across the life stages. We did attempt to make a universal lexicon so that everybody, whether or not you studied frogs or algae, could understand the same concept. And then we also came up with an experimental design that does use a genomics approach, but is not limited to a genomics approach. So just to start off, just to get on the same page, we defined a life cycle as sort of a conceptual model of the changes an organism goes through between, say, being laid as a birth and then birth at the same point in the next generation. And just to give you an idea of the diversity of different life cycles that exist, this is an image we use in the paper that shows four different life cycle forms that can be found in your standard kelp forest habitat. So there's an otter, urchin, crab, and kelp. And of course, there's tons of others. We just highlighted these four, to show the diversity that can exist within a single habitat. And of course, what we were interested in was a complex life cycle. And this is a form of a life cycle where those changes in development occur in discrete stages and typically include very complex morphological, physiological, ecological changes. It can include organisms that go through a metamorphosis, but is not, a metamorphosis is not required to have a complex life cycle. And I love this art by Bela Arrieta. And so to go through this, I am going to show my animal bias. I'm going to use a dragonfly. It's not a frog, which is what I typically study. So I am expanding my boundaries a little bit, but I'm going to talk about this conceptually using this as a heuristic. And so what is interesting about having a complex life cycle is even though an organism is going through these very different stages of their life, they are all connected by the same genome. And so the question is, or the thing that kind of bubbles up is, well, your adaptive potential, therefore, depends on your performance, not only within stages, but also across stages. And so can traits evolve independently? Will the traits that evolve in one stage affect the traits in another stage? These are all sort of lingering questions that we really don't have an adequate answer for. Very few studies capture the whole life cycle. This is some data I just pulled from our paper to show that most studies focus on one or two stages, with very few extending beyond 2 stages or capture the entire life cycle. Or we're sort of relying on these quantitative genetics approaches, which involve genetic variants, covariance matrices, which are somewhat involved and difficult to experimentally to run. So we were trying to think of a different way to do this that is experimentally tractable within a single generation of a life cycle. So we came up with a new framework to talk about these things. I want to start off that there are 3 potential ways that selection can act across. And we start each of these terms with the word ontogenetic. Ontogenetic means that it's moving developmentally through the life cycle. So going from egg, developing into larvae in this case, and developing into adults after that. And so there's one situation where you can imagine that selection is acting on fitness components at this larval stage and have an effect here, but then are neutral. Those fitness components are neutral at another life stage. And we call that adaptive decoupling. So this is essentially where the impacts of selection are isolated at a single stage. There's another potential scenario where the actions of selection on fitness components at one stage affect that stage in one direction, but say in the next stage, they affect, they're moved in the, those fitness components are moved in the opposite direction in the subsequent stage. So we call this antagonistic pleiotropy because the effects of selection are moving against each other or counter to each other across the life stage. And then finally, there's the situation where the effects of fitness at one stage, the fitness components experiencing selection at one stage experience selection in the same direction in the next stage. And then we call this ontogenetic synergistic pleiotropy. So these are 3 general ways that selection could be acting on traits across the life cycle. And when I say traits, again, I'm showing my bias. This could also be alleles, all sorts of different phenotypes. And this framework allows us to formulate new predictions that then we can build an experimental design. So for ontogenetic decoupling, the traits or alleles that change frequency in one stage don't change or should not change in frequency in the other stages. So this is sort of beginning to develop hypotheses about how these things should appear under an experimental setting. Antagonistic pleiotropy should show traits or alleles that change frequency in one stage, say increase in frequency, but then decrease in frequency in the subsequent stage. And then finally, you can imagine synergistic pleiotropy. They increase, traits or alleles increase in frequency in one stage, for example, and then increase in frequency likewise in the same direction in another stage. However, not only is the environment likely to change, but the relationships among traits could also change too. So then what we needed was a way to define these relationships in the current environment, which I which we call control. Of course, we all know that that truly doesn't exist, but in the current control environment. But also we needed a way to define these relationships in a future or experimental environment. And this could be whatever climate change, environment change you're interested in, salinity, temperature, acidity, whatever. And so what we came up with was this experimental design where you rear an organism through their life cycle. So from stage 0 to stage 1 to stage 2. And so it's just a single generation where you rear half or part of the individuals in the control or current environment and half in an experimental environment. And for our purposes, because we were interested in allele frequency and a more genomics approach, we use delta P to signify these changes in allele frequency as the sort of the thing we're looking at through the life cycle. And so what this shows as we rear individuals from stage 0 to 1 to 2 is if we look at the allele frequency across stages and compare stage 1 to stage 0 and stage 2 to stage 1, then we can get an idea of these relationships of antagonistic pleiotropy, synergistic pleiotropy, decoupling in the current environment. And really, we just get the way that allele frequency changes in the experimental environment. So from stage 0 to stage 1, stage 1 to stage 2, then we can also get an idea of how allele frequency changes across stages in the experimental conditions. And that can tell us whether or not those relationships are changing. Let's say there's a situation where we have antagonistic pleiotropy in the control environment, but in the experimental environment, it switches to become a synergistic pleiotropy situation. And so at each iteration of the life cycle, you can also compare allele frequency between experimental and control conditions. And that tells you how selection is changing allele frequency in the treatment. And we pair those with stress tests to get an idea to link these allele frequencies to fitness or performance. And then of course that can happen at each stage of your life cycle. And of course this is adaptable to whatever organism and how many stages that organism has. And so the caveats of this approach, it's a solid approach, but of course it's power hungry. So as you can imagine, we did a power analysis. Katie did this power analysis using simulations. And what we're showing here is sequencing depth versus the sample size to detect an allele frequency change. So right here is a 0.1. So that's like a very small or modest allele frequency change. Sorry, I don't think you can see my mouse. I'm pointing at the 0.1 on the x-axis here. And then you can see that as here on the left side, we have 100 individuals, and then sequencing depth increases as you go down the plot. And then on the right side, we have 10,000 individuals with sequencing depth going up as you go down the plot. So as you can imagine, as we know, more power comes when you have a greater sequencing depth with many more individuals. But you can attain power depending on what effect size in terms of allele frequency change you expect. And then of course, all the different caveats of your study system. But that does mean that that taxa with really large numbers, like marine invertebrates, a lot of marine taxa that have like really large clutch sizes or offspring, are amenable to this approach. And then of course, none of us can afford to sequence 10,000 individuals. So using techniques like pool-seq, where you pool individuals into a single sample, help make this sort of large scale design much more possible. But again, we're not limiting to that approach, since of course, the whole point of this project was to think about these things very broadly, not restricted by taxa. So it is a flexible design with consideration, of course, for your organism's natural history, the sample sizes that you can expect to get. Of course, cost is always an important thing. Your effect size, in this case, the allele frequency you expect to see. And then there's also the potential for confounding effects like density. Anytime you're applying a stress where you anticipate mortality, you're also going to be reducing density. So there needs to be some interest in determining how those changes in density might affect the allele frequency or the outcome. And so what we ended up coming up with was not so much an answer to this question, because we quickly determined that right now we don't have an answer to this question of how does having a complex life cycle affect adaptation. There's not good data to really make solid conclusions, but we did come up with this way to study it, and we hopefully came up with this way to talk about it that can really advance this field. So the way we talk about it and the way we study it, we've got this framework now, hopefully will be used. I know it's a mouthful. We have an experimental design that can be adapted to different taxa systems and can be complicated depending on your ability to say incorporate a dose response design. And then we also encourage everyone to ask themselves as they think about these questions, will an expert in, say, algae understand this terminology? Will the expert in jellyfish understand this terminology? And always define your terms. And if you need to, use a glossary. Again, I won't belabor it because there's a panel on that later. And then hopefully we can come up with an answer to this question, because it's a really important one. Some 80% of taxa are estimated to have a complex life cycle, so it's likely to be important in terms of future persistence. And so with that, thanks to all my co-authors, the workshop planners have done a phenomenal job, Joanna and Katie. Of course, the RCN, I was funded through the RCN, so I'm very grateful to them and the NSF for providing me funding as well as for this workshop, and I would love to answer some questions.
Thank you very much, Molly. So we'll open it up to questions, and I'll say, people on Zoom, please type your questions into the chat and they will be read aloud here. So I encourage that to happen. So are there any questions from the room? All right, I'll run this over to you. And maybe just state who you are.
Hi, Molly, this is Nina Terkelsen. Thanks for a great talk. So at this point, are there any empirical data? Is there any species for which you have, maybe not perfect, but whoops. Oh, okay, where we have data that at least somewhat informs this framework that you've laid out?
There are some data. Most of them have relied on a quantitative genetics, so the genetic correlation approach.
Sorry, Molly, we couldn't hear the beginning of your answer. Would you mind starting over?
Sorry, sure. There are some data they typically have used a quantitative genetics approach to study this. These efforts are reviewed. There's a study that does a really great job of reviewing this using a quantitative approach. It's Collette and Fellous, Fellous. And so if you're interested in sort of understanding that, check that paper out. Off the top of my head, I can't think of any authors that specifically do this, but I know that there's like a smattering. It's just not really, there wasn't a design that was applicable across taxa, which is sort of where we sort of kept running against this wall of, well, how does somebody who doesn't study that taxa understand this question?
Great, thank you.
There are other questions from the room or from Zoom? Okay, I have actually a question, Molly, which is, we all well, maybe not all of us, but many of us have our pet organism that we're really attached to and we know a lot about. How much do you think that there is a chance of coming up with like a really broad framework that encompasses all the different life history strategies? Or is it really that we need to be focusing on each different type of life history strategy independently in this hope for like the huge framework? It's just there are too many and that there is no real overarching solution.
Boy, this is, I'm sure, a philosophical question that everybody has an answer to. My personal, I think about this all the time, and the answer is that I don't have an answer, because the more I learn about things, the more specific to that taxa it seems, but then you can always zoom out and see commonalities that apply across taxa. So the answer is yes to both. I think, I don't think I'm going to be very, elegant in my answer because it's 6:30 here and I haven't finished my coffee, but I do think that we have to, I think that it's important that we experimentally use the taxa we know, because natural history is really important. And if we have expertise on the natural history of that organism, we can do a better study. And I also think it's really important to continuously zoom out from your study organism so you don't get too myopic, and you can sort of look for these common threads that exist across different life cycles where they do exist. That probably wasn't a great answer, but the answer is I'm always a fan of people doing the work on the animals that they know, while also trying to, do the impossible task of read widely and find commonalities across taxa and systems.
Good answer and very thoughtful, and I appreciate that. And it looks like there is a question on Zoom, so we'll have that read aloud for those of us that can't actually read the screen there. So, Stephen.
Yeah, so we have a question from Serena. It says, really great talk. I know this complicates experimental approaches, but it seems selection pressures are likely to vary over time and thus life stage. Did your group think about ways to tractably increase the complexity of the experimental design? How much does adding variables reduce power?
I'm not exactly sure how much, I couldn't give you a number, but it does reduce power to increase the complexity of the experimental design. With that said, we purposefully put this super simple design in the paper so that it could be complicated, just so that the basic bones of that experiment were communicated, while you can expand it to include more life stages. You could expand it to include, say, a dose response experimental design, add more experimental conditions. But with that said, yes, it should reduce your power. So there should be some consideration to your ability to detect small changes, if you're interested in small changes, as you increase the complexity of your design. But yes, you can increase it. Yeah, effect size, the all-knowing effect size, yes.
Is there another Zoom question?
Following up and saying thank you still depends on effect size too, I guess.
Okay, great. Well, I think with that, we'll give Molly a big congratulations on a great talk. And very happy to see the output from that working group. It's really exciting to see all these results being presented and coming together. So thank you. All right, so we'll get Mark set up here. Okay, great. So next up is Mark Bitter, who's going to be talking about fluctuating selection and global change.
Right on. Thank you very much. And just first want to thank the RCN organizers, the steering committee, the PIs, the workshop organizers, and also Scholes for hosting us here. And then this just went black, so we'll try to catch. No.
This is actually perfect. We were 1 minute early. But right on time. So that works.
Okay, cool. Yeah, so once again, thanks to all. Today I'll be discussing fluctuating selection and responses to novel climates. This came out of the first workshop. Up there is our group's first scribble that was on the back window. And then to the bottom left is, the citation from this, what I think is a really nice paper that came out of it in the special edition. And then above that is the list of my really incredible co-authors that I really learned a lot from. So to dive right in, cool. And kind of like a parallel to the last question that was asked, I think we all know that just assaying a single species vulnerability or adaptive capacity to a single climate change stressor is pretty challenging. So it seems like a pretty insurmountable task to assay every species vulnerability across every stressor and combination. And so therefore, it might be useful to try to understand just general environmental features about some population's natural habitat or the environment that it evolved in that in some way predicts its response to expected changes in mean conditions. So this would look like taking into account some contemporary environmental time series data or some time series from the environment a population evolved in, combining it with climate change projections and making some prediction about how biology, phenotypic variation, or genetic variation might change as a function of the environment. And so our group really addressed this specifically within the context of fluctuating environments and how different characteristics of a fluctuating environment might influence a population's likelihood of persistence under novel climate conditions. And so first, I'm just going to go into this concept of climate novelty, a little bit about how it's quantified, and what the expectations from that literature are. And so we now kind of expect that climate change to induce abiotic conditions that are not found at present by the end of the century. This is the term novel climates, and it's a metric that indicates how far a particular location in space is expected to travel in a multivariate environmental space at some point in the future relative to current conditions or reconstructed data. So for terrestrial systems, one might take into account temperature and precipitation, for marine systems, temperature, salinity, carbonate chemistry, and just ask whether a particular grid point is going to experience some combination of environmental values that exceeds what the current contemporary data holds. And so people have done this in marine and terrestrial systems. Here's a figure from Katie's paper. Sorry, this is a little bit small, but the shading, we're just looking at the world's oceans right now. The shading corresponds to the Mahalanobis distance, which is this multivariate distance metric. And for each grid point, it's just plotting how far expected conditions are expected to transcend into novelty. And the general trend is that we see the greatest degree of novelty in the tropical and the polar regions. And one can do this under different emission scenarios. So this is just different emission scenario, and we see the same trend whereby the tropical and polar regions are expected to exhibit the greatest degree of novelty, but just a bit more drastic scenario. And then one can also do this with reconstructed data. So this is reconstructed data from 1800, same trend, and we've probably already observed emergence into novel environments. When this is done in terrestrial habitats, we get the same picture. And so the main takeaway from this body of work is that this projected degree of novelty is relative to the magnitude of environmental variability at a location. And so regions with kind of low amplitude environmental fluctuations, most notably the tropics and polar regions, are expected to experience the greatest degree of novelty or the soonest time to emergence of novel conditions. A lot of the papers then kind of make some speculative inferred biological implications, just suggesting that populations and species living in these either low latitude tropical or polar regions are probably at the greatest ecological risk of global change because they're going to experience climate novelty sooner. However, one potentially limiting assumption of this approach is that it really discounts how variation that's maintained within populations might facilitate their persistence under climate novelty, irrespective of when that novelty occurs. And so what we really wanted to ask is whether there's other components of a fluctuating environment that might influence if and how a population persists once novelty arises. And so to do this, we took a deep dive into theoretical literature. And we first, before thinking about a global change scenario or climate change, we wanted to understand how environmental fluctuations shape the phenotypic variation in natural populations. And we focused on quantitative genetic theory models, and particularly this norm of reaction approach. And I think the norm of reaction is first coined by Via and Landy. And for this, I really want to highlight Scott Burgess in our group, who's really an expert and has contributed to this quantitative genetic theory for kind of walking us through this. So to just go through the models, generally the normal reaction, there's different forms, but with a very consistent parameters. And so a reaction norm just describes how the phenotype of a genotype changes as a function of time. And I'll walk through each of the parameters and kind of draw it out here. So the A parameter is the intercept of the reaction norm, and this could be the phenotype in the reference environment. So here the reference environment might be standardized or normalized to some 0 value. And it's also referred to as the breeding value. And it's important to note that in all the models, there's some additive genetic variance in each of these parameters. And so here we're just looking at variance in breeding values for genotypes J, I, and K. The next parameter is B, which is the slope of the reaction norm. And this just dictates the degree and direction of a plastic response given some shift in the environment. So to fill out the reaction norm for genotype I, it might look something like this. And just to be super explicit about this, this could be changes in shell size as a function of changes in temperature. And then similar to breeding value, additive genetic variance, there's additive genetic variance in reaction norm slope as well. And so finally, how selection works in the models is for each value of the environment, there's an optimal trait value, usually denoted theta. And so theta for environment T+1 might be right here. And fitness is then determined as just the distance of a genotype away from this optimal at the time of selection. And finally, we now need to think about how the environment is modeled. And once again, just like with reaction norms, the general form can vary across models, but the parameters remain the same. We chose this form from Rio-Coulain et al. 2009, whereby the environment at any time step is a function of the environment at the previous time step, and then these rho and omega parameters. Omega dictates the variance around the mean environmental state. So this is really the amplitude of the fluctuations. And then rho is the autocorrelation of the environment between two time steps. So how correlated the environment is between time step T-1 and time step T. And this ultimately dictates in the models the predictability of the environment. And I'm going to go a little bit into environmental predictability now. It's kind of this concept like fitness, whereby like it's just a human construct, but something that we think might be biologically meaningful, and we might assign values to try to understand how evolution works. And so from a biological perspective, this environmental predictability is the correlation between some environmental cue sensed by an organism that influences the development of its phenotype, and then the optimal phenotype when selection occurs. So consider this muscle example where there might be variation in muscle shell size that might have kind of some plastic component to it, and this selective environment, which might be the presence of sea stars. And the sea stars might selectively harvest kind of the longer muscles that have maybe thinner shells, leading to some fitness benefit to smaller, thicker shells in the presence of the sea stars. So then consider the scenario whereby the presence and absence of sea stars is a fluctuating selection, but the sea star might emit some cue that is indicative of its presence. And this cue might be able to be incorporated by some little mussel that's looking to settle and start growing its shell. And then we might get some pattern that looks like this, whereby in areas with no or few sea stars, we have larger mussels, and in an area with many, we have smaller mussels with thicker shells. And so this just corresponds to some kind of predictably fluctuating sea star environment, because the Q is really predictive of whether or not sea stars are there. And one really important thing to note, just from a biological perspective, is that, you know, whether or not we can quantify that this Q is predictive of sea stars being present, what's really only biologically meaningful is if the organism with which this exerts selective pressure is able to sense that Q and alter its phenotype in a timely manner. Okay, so now I'm going to go through really what the theoretical predictions are. And once again, we want to take into account some environmental time series data. But in this case, we want to think about a range of time series that alter orthogonally in these relevant parameters, so the amplitude and predictability, and act how these parameters interact to influence reaction norm evolution. For this, I want to highlight Katie, who did the environmental simulations here, is really supportive and great help on this work. And so we're going to think about how each of these kind of environments of evolution might shape how phenotypes change as a function of shifts in the environment. So deviations away from this mean environmental state. And all in reference to this optimal phenotype, which here is set to plus 10 units of the current environment. And we're going to be looking at just the population mean reaction norm. So ignoring kind of variation within population variance, and just asking how each of these environments of evolution might lead to kind of a reaction norm with different slope or elevation. And so what's important to also note is that we also want to think a little bit about these scenarios down here, which are conditions that might exceed kind of the contemporary observed distribution of environmental values, so novel scenarios. And we really don't know what happens here, how phenotype or how genotypes respond when they're exposed to some environment they've never seen. And so models usually consider two scenarios. One is that the reaction norm is just physiologically continuous. So that slope of genotypes I reaction norm might be 0.5, and it just continues as it experiences continuously novel environments. The other is this release of cryptic variation, where there's this disconnect between what genotypes do pre and post exposure to novel environments, leading to these non-linearities. Ultimately, I kind of reference Cameron Galambor's perspective on this in 2007 that goes through in a bit more depth these different scenarios. Okay, so now, once again, each color of the reaction norm is going to correspond to one of these different environments, and we want to kind of systematically compare what happens when we hold different parameters constant and alter just a single parameter and compare reaction norms. So when we compare this low amplitude, low predictability, the dark blue, to this low amplitude, high predictability, so we're just asking how increasing the predictability alters this reaction norm, we see a picture that looks like this, whereby the population that evolved in the predictably fluctuating environment has more plasticity and more effectively tracks the optimum. And this is because these predictable environments provide reliable cues for organisms to track that optimum. And this is like the corollary being that unpredictable environments cause organisms to alter their phenotype in response to unreliable cues, and then it sets them off the optimum, and there's a fitness detriment to that. And ultimately, totally unpredictable environments in the models lead to these fixed phenotypic strategies like bet hedging. So the next question is, what happens when we keep the predictability constant, but alter the amplitude, so increase the amplitude of fluctuations? And in this scenario, we do see like an elevation of reaction norm slope in a population that to some degree is tracking the optimum. And this is because if predictability is held constant, increase in the amplitude leads to increase in plasticity. But when we superimpose kind of this still low amplitude, high predictability scenario, we see the population is more effectively tracking the optimum. And this reason to that is that the influence of amplitude is really secondary to that of predictability, and amplitude actually just determines the strength of selection acting on plasticity, which dictates the rate at which the population evolves to some optimal level of plasticity, which in turn is actually set by predictability. And just filling the figure out fully, this is the high amplitude, high predictability scenario. And so next, what we want to do is ask how these reaction norms then translate to potentially the fitness of the population. And as I mentioned before, the fitness is just determined as the distance away from the optimal phenotype when selection occurs. This figure kind of is a little bit misleading because these deviations in the environment are more dramatic for these low amplitude scenarios. So this figure has a rescaled x-axis that just represents how fitness changes as a function of the environment with respect to the number of standard deviations away from the historic environment. And when we do this, we get a picture that looks like this, whereby while we can't really here disentangle the effects of amplitude, we just see this general pattern whereby fitness decline is less rapid for the populations that evolved in these predictably fluctuating environments, notably the light blue and the light green. And what this tells us is that at least in the short term, these evolving in a predictably fluctuating environment facilitates persistence when exposed to novel conditions. And so therefore, at least in theory, population sensitivity to global change might not be wholly dictated by just the amplitude of fluctuations the populations experienced. So the next major question is, how do we actually test these theoretical predictions? Well, we want tests that really disentangle systematically the effects of amplitude and predictability. So we'll just highlight kind of what our group thinks are necessary criteria to do so. First is we really need to look at studies that are either collected from or evolved in environments that vary orthogonally with regard to amplitude and predictability. So a scenario like this that I mapped out. And then experimental levels really need to be chosen with regards to what's observed. And we need to look at experimental levels that both are within kind of the observed range, but also systematically look at deviations into novel environments. And then finally, we need to both kind of assess reaction norms and how these reaction norms may impact some fixed fitness or fitness proxy. And so we were really interested in how well empirical studies have addressed these theoretical predictions. And so we did a bit of a literature survey and specifically focused on marine systems and how well they met these criteria or some relaxed criteria. Oops, sorry. And this focus on marine systems was in large part a result of the goal of this group, but also because marine systems generally have conditions that dominate over longer timescales, have greater autocorrelation and potentially predictability. There's also a large range of biologically relevant fluctuating selection pressures in the system with which we can test theoretical predictions. And finally, there are climate change stressors in marine environments that aren't present in terrestrial systems. And for this portion, I want to highlight Juliet Wong, who really led kind of this challenging investigation into the literature, because defining some of these criteria and how specifically we can assess if a study met them or not is a really challenging venture. And so just to quickly highlight some of the main results from this, we're going to look at the number of studies that either took historical measurements, made experimental manipulations, measured reaction norms and fitness, looking at the raw number in the bottom left, and then it disentangled into whether the study was intergenerational, transgenerational, or experimental evolution on the right. When we do this, we get a picture that looks like this, with just some summary stats being that 60 studies met our basis for inclusion, none compared populations that were collected from or involved in environments that just demonstrated all these 4 amplitude predictability scenarios that we need to kind of disentangle their effects on reaction norms. And while many did just quantify reaction norms and fitness, few related them to historical or manipulated levels of amplitude and predictability. And few really chose experimental levels that we know were novel for the population of choice. And I just will kind of draw your attention to the paper for more specific examples of studies that have done this well from both marine and terrestrial systems. Okay, and then avenues for some future research. I think our group kind of converged upon the idea that we really need direct tests of theory to answer like these two major outstanding questions. What happens to reaction norms in novel conditions? Is there this simple linear extension of reaction norms? How much does plasticity actually facilitate persistence in novel conditions? And then really disentangling the effects of the amplitude of fluctuations and predictability in shaping the degree of plasticity in natural populations. That's all I got. Once again, thanks to my really great co-authors, the Evolving CPI Steering Committee workshop organizers. Thank you very much. Yes, Jamie. Oh.
Hi, this is Jamie Pringle. I was wondering, in the classic sort of climate change literature, you find larger changes as you go to higher latitudes, larger changes at colder times, either nights or winters. And I was looking at your prediction that there was more change in the tropics. Is that because it's normalized by the annual fluctuation? Oh, well, I'm just going to add on one little bit to that. It strikes me, if you look at life histories around here, most creatures are limited not by an overall measure of the local climate, but either a minimum winter temperature or the time of frost, that they're more sensitive to one season. So it's the variation against the average for that season. Like my crocuses don't care how cold it gets in the winter. They worry about the end of the frost in the spring. So is it really appropriate to say that there's less change here when most species are sensitive to just a particular season's climate?
Sorry, Mark. I want to make sure everyone can hear your answer.
So I'm just going to try to restate some of these points, which is that so the novelty metric, it is standardized by the degree of observed amplitude of fluctuations. And so I think your first point was that we expect the greatest magnitude of change in higher latitudes, and you're asking whether the novelty metric though is standardized, but by the observed range contemporarily seen, and it is. And the second question is though that this novelty metric is kind of this net sum of the annual variability seen, but in fact the sensitivity of organisms is really dictated by like a particular event within the year, within a season even, and therefore how important is, I don't know. I think that's, the novelty metric, as I understand, Katie, if you can agree, it is like kind of this net sum annual effect and deviation from that. So it does not take into account like the more fine scale events that might actually dictate sensitivity. And those probably are quite biologically important. So I agree. Yeah.
Thanks. Super cool talk, Mark. You talked a little bit about how fitness declines vary between populations that evolved in predictable and non-predictable environments. Did you look at how changes in predictability affect those declines? Or so a population from a predictable environment that's exposed to increasing levels of unpredictability?
Yeah, you could see that like just by comparing the same color graph, which would be just comparing like, for example, the light green to the dark green or the light blue to the dark blue. So this is where amplitude's controlled for and predictability is manipulated. And these have it, you can like the comparison for just the environment then is the same. They've observed the same amplitudes, the same amount of change, the same amount of novelty, and you see this kind of slower decline in fitness as you deviate away from the mean. Thank you.
Hi, Eric Satka. That was a really nice talk. I see that when you put the empirical studies together, you came up with a list of studies that had approached this, but I guess I didn't see that whether those studies actually supported your theoretical predictions or not.
Yeah. So at least like in that regard, you would really want to, yeah, they would need to be kind of one of these subset of studies that met kind of both looking at reaction norms and the evolution environment of evolution. And there are very few studies that did this. One group's work, like Louis Miguel Chevin in France, they've developed a lot of theory and gone really explicitly into testing it. And they have shown that unpredictable environments lead to depressed plasticity in these microbial experimental evolution experiments. Lead to depressed plasticity. And so, yeah, just see if you can see the paper, we kind of highlight some of those, what we think are currently the best examples that have effectively tested theory and whether or not they've kind of met the predictions. Okay.
All right, let's thank Mark for a great simulating talk. Katie, is there a plug anywhere? All right, great. So next up, we have Jennifer Hoey, who's going to talk about whether or not spatial and genomic estimates of connectivity agree. And I think you will need to. Yep.
All right. Hopefully everyone virtually can hear me. Good morning, everyone. So I'm presenting on behalf of my working group. So Alli Cramer here in the audience was my co-lead on this working group. And then other members include Tara Dolan, who was a former UC Santa Cruz postdoc and is now at the Massachusetts Division of Marine Fisheries. Remy Gattens, who's online, I believe, and is a postdoc with Katie. Jason Toy is a Santa Cruz PhD student. Jordan Chancellor is a PhD student at the University of Southern California. And then we also worked with Eric Polkovacs, who's a faculty member at Santa Cruz, Carlos Garza, who's at the NOAA Southwest Fisheries Science Center in Santa Cruz, and then also another Santa Cruz faculty member, Roxanne Beltran. So I have some technology associated with my presentation, so hopefully it works. If I can advance the slides. Okay, so how many of you have used Pol EV before? Great. Okay, so here's a warm up. So in case, hopefully the instructions will appear for you. But if you're on the Wi-Fi, you can go to polev.com/Jennifer Hoey. Or if you have cell service, you can text Jennifer Hoey 504 to 22333. I had tested this before and it doesn't seem like the instructions are showing up for you. Yeah, it's Jennifer Hoey 504. Sorry. Hoey. Yeah. Cannot see the chat. Okay. Oh no. Okay, I'm sorry. It is not. Because I can see it on my screen, but it's not projecting to you. Um, uh, okay. I think I'm almost there. No, now I have to find. All right, I am having troubles. Unfortunately. Yeah, not anymore, but. Okay, I think it's too challenging to figure it out on the spot. So unfortunately, I'll just have to show you the responses later. Let me get back to the presentation. Okay. So you guys responded. I could see it on my computer, but it wasn't projecting the right screen for some reason. So I do have some additional. Oh, okay. Maybe this one will work. So it's the same link. But it is also not showing your responses. Okay, but most of you, 52%, are identifying as both ecology and evolution. 56. And now up to 60. Okay. And then the question I'm really interested in getting from you guys is, this will be hard to see, so I will also share it later, but in 1 to 3 words, I'm interested in how you would define population connectivity. So I'm seeing gene flow, a lot of gene flow, migrants per generation, admixture, migration, words about dispersal, sharing, generation, numbers of things, inbreeding, but predominantly the largest word on this invisible word cloud to you is gene flow. And so the original goal of our working group happened due to a chance meeting between Alli and I during the summer of 2020 when we met at a postdoc kind of happy hour at Santa Cruz. And we got talking and we realized our backgrounds were really different, but we were interested in some similar things. And so our original goal was to try to integrate across subdisciplines to gain a new inferences and specifically about connectivity. So our original question was, how are dispersal patterns reflected in genomic population structure? And similar to, well, first we developed an initial partnership with another working group, which we'll hear about later, the Predicting Diversity and Divergence in the Sea working group, because they have a bunch of population genomic data that we wanted to take advantage of. And then our initial idea was to select different species with different movement ecologies. So species that exhibited very different, for example, degrees of movement, degrees of philopatry, degrees of homing. And we wanted to learn the SLIM modeling framework to simulate these species movement ecologies and then compare the simulation output to the population genetic data from the other working group. But similar to the life history working group for Molly, we quickly ran into challenges of trying to communicate with each other. And so we did develop this initial partnership with a sister working group and had some conversations about sharing data. And then we tried to tackle the second and third bullet point of picking species and learning SLIM simultaneously. But where we really got stuck was on the second bullet point of deciding which species to actually choose. And this was because ultimately we couldn't agree on which species characteristics were most important. And this was because I think we were tending to focus on our various methods that we were most familiar with. Mostly it fell into 2 categories, either genetics or using tags to track individual organisms. And then we also quickly realized as we were talking to each other that there were differences in assumptions based on our background and our methods. And then when we were looking at population connectivity in literature, there were many, many definitions, and then which made it even more confusing about how to define different species characteristics that we should even be looking at. And so I think the way that we were previously looking at connectivity, it was ultimately like looking at 2 sides of a coin. One of us fell on one side, the others of us fell on the other side. And we couldn't really have coherent conversations with each other because even though we were ultimately talking about very similar things, we were seeing it from only our own perspective. And so the way we're trying to tackle this was also with a conceptual framework that tried to integrate our different perspectives. So rather than viewing connectivity as two sides of a coin, viewing it more as ripples or more as a continuum. So in our generational framework, we have time as a continuum. And then on the most recent time scales, we have ecological time scales. And on longer time scales, we have evolutionary scales. And so if we think about this in terms of ecological population connectivity on the most recent time scales, here's an example. So here in this photo, we've got a Google Earth image of elephant seal tracks. So those white squiggly lines in the ocean are tracks of individual elephant seals that have been tagged. And then it's a little bit challenging to see, but there's a yellow dot here in Mexico and then a blue dot here in California. So if we imagine we tagged elephant seals at both of these places at some point in time, there's the elephant seals. And then at another point in time, we observe these same animals in the same place. So from an ecological population connectivity perspective, we would conclude that these two populations are interacting. They're in the same space at the same time. So for ecological population connectivity, it's the movement of organisms across the landscape. And assumptions of this are that these organisms are occurring in space and time, that the individuals that you're tagging are reflective of their population, and that also their present behavior reflects their past behavior. And so this can have downstream impacts for things like population persistence, potentially observed population structure that you might see, and other ecological processes like predation and competition, and then also the flow of nutrients, and all other examples include transmission of disease and also culture. So if you look at population connectivity from an evolutionary perspective, if you can contrast it to these same elephant seal tracks. So this is a hypothetical example for elephant seals, but probably many of you have seen a structure or admixture plot similar to this. But if you haven't, each individual vertical bar represents an individual, and the proportion of colors within the bar represent the proportion of ancestry to a putative population. So I just made up this example for elephant seals, but so it's very clear cut that there are two populations, one blue and one yellow. And so from an evolutionary perspective, from this plot, we see that there's no admixture between the populations. Although if you were comparing it to the ecological perspective, these individuals were interacting in space and time. And so from an evolutionary perspective, we're thinking about the degree of gene flow. And some assumptions of this include that there's physical proximity of organisms or gametes at some point in time to make reproduction possible. And that also these individuals that you're sampling are also reflective of their population. And then the degree of gene flow can also impact things like genetic diversity and local adaptation to particular conditions, and also effective population size. So placing these onto our continuous time scale, we decided we wanted to think in terms of generations. So within a generation is our shortest time scale here. And so for this time scale, you can use occurrence tags, so radio transmitters or other forms of physical tags to track individuals between populations. And so here if you're tracking them with physical tags, you're observing ecological connectivity. And there could potentially be evolutionary connectivity, you just don't know. So occurrence tags relate individuals within their particular generation. And then on longer time scales, 2+ generations, we can think of that falls on the evolutionary continuum of the spectrum. So here you can use tools like population genetic tools to look for shared ancestry among individuals and populations. And so we're inferring evolutionary connectivity based on these markers, and then there could also potentially be ecological connectivity. So if we sampled this elephant seal here, who just turned green, and looked at its ancestry, we might conclude that it's an admixed individual from a yellow and a blue population. And then on intermediate scales, we're calling these intergenerational tags. So on a scale of 1 generation, you can do parent offspring analyses that reveal the ancestry, or you can use non-genetic tools too, to track relationships between parent and offspring across 1 generation. So at this time scale, you're inferring that there's ecological connectivity, and that you're also observing evolutionary connectivity because you're observing that direct transmission of alleles between parent and offspring. So many of you said that you were identified as belonging to ecological and evolutionary, sort of investigating those sort of questions. And so we do too within our working group. And so we wanted to synthesize how using this generational framework might help you think about and help us think about feedbacks between ecology and evolution. So the question is, how might ecological connectivity impact the mating pool? Because that's what matters for evolutionary connectivity of a single generation and downstream evolutionary connectivity. So if we think of a mating pool of elephant seals here in blue, and then something happens where the availability of mates increases. So in this scenario, all things being equal, you'll probably get increased gene flow and increased genetic diversity, but you might potentially decrease local adaptation if there's an influx of new genes into a particular location. And then the opposite can also happen to lower the size of the mating pool. So if something happens where many of the blue seals are gone, you decrease the availability of mates, which will, all things being equal, also decrease the amount of gene flow and genetic diversity, which maybe over time might increase local adaptation. And so continuing the cycle, how might evolutionary connectivity impact an organism's ecological connectivity? So if you have some allele which controls some ecologically important phenotype, and then also the current abiotic or biotic conditions relevant to that phenotype, you might get selection that will then alter the distribution or behavior of your particular population of interest. And if this happens, and there's a different distribution or behavior of your population that changes the way it interacts with a different population, then that could potentially impact ecological connectivity by changing the number of mates in that mate pool. So by using our framework that we constructed to help us talk together, we came up kind of with this flow chart of how eco and evo population and connectivity might cycle back on each other. And so ultimately what our group kind of produced, so far is that population connectivity exists along a time continuum that we've defined by generations. And this generational framework provides a structure, and I think it helped us, and hopefully it's helpful for others too, for synthesizing information about population connectivity and just thinking about it in a slightly different way so that we could talk to each other. And then ultimately, what our main goal was to integrate across our various disciplines and backgrounds so that we could have a more holistic understanding of connectivity in natural systems. And so where we stand right now, we'd love some feedback on this framework from you guys. So you can come talk to me or Alli at this meeting. But we're trying to prepare this conceptual paper for publication. And then we ultimately still want to look at our original question too. So we're also seeking collaborators and other funding opportunities to try to complete this work. And hopefully we can use the framework that we made to help better guide us in our next steps for this. So with that, I'd like to thank my collaborators and then also the NSF Evolving Seas Committee. We got a bunch of good feedback from Joanna Kelly and Peter Dudley. And then also Danielle Dubé helped us with the elephant seal scientific illustrations. So with that, I'd be happy to answer any questions.
I think there's one Zoom question. We'll have time for one question to keep on time. We'll get that Zoom question.
Sure. So the Zoom question is from Catherine Silliman, and it says, what if you have a marine fish species that reproduces offshore, but spends most of its time in nearshore estuarine habitats? How does that fit into your framework?
Yeah, so I think you can try to answer this question using the framework by looking at different pieces of the life history. So maybe you can track individuals within a single generation, and then you can also look at population structure questions. So I think our framework is usable for things with different life histories. It's just focusing on, it's like knowing about the life history, but then also kind of using that to tackle different aspects of the connectivity question. And multiple studies might need to be done for that too. But by hopefully focusing on the different generations, you can gain a better picture of how this marine fish is, what it's doing.
All right. Jennifer again. All right. And so the last speaker of this session is Matthew Sasaki, who's going to talk about surf and turf.
Oops. Oh no. Let's see. My Zoom just updated, and so all of this is gotta leave my meeting and rejoin. Oh no. Okay. I think that's good. I'll go back here. Yeah, it wouldn't let me share my screen. Sorry. Okay. Am I still? I think I'm muted. Okay. Perfect. Thanks. Again, my name is Matt Sasaki. I'm a postdoc at UConn, and I'm here to represent the Surf and Turf group, which in hindsight is maybe not super informative about what we're going to be talking about. But our group, so myself, Jory, Morgan, Sarah, Cynthia, Alli, Seema, Andrew, and Brian have been working for the last couple of years. We started at that table right there at the first synthesis workshop, and put together some cool products on comparing patterns in local adaptation across different habitats. It's been an incredible experience, and so I'm so happy to have been able to work with and learn from all these people. And that's something that would not have come together without the support of the RCN. And so I also want to thank the RCN organizers for everything they've done to give us the support we need to do this work. So when we started thinking about project ideas, we were really interested in how local adaptation might interact with climate change to affect something like population vulnerability. And right away, we started thinking about thermal limits, or the maximum temperature that an individual can withstand. So this area of a performance curve. And while this is the most extreme sort of effect that an environment might have on an individual, right, mortality being sort of an ultimate endpoint, these thermal limits do provide boundaries to performance and therefore have influences on ecology, biogeography, and all these other things. And so we wanted to focus on local adaptation of thermal limits and how that might vary. Luckily for us, there's been a lot of work done already on patterns in thermal limits across species. And some of you might be familiar with the GlobeTherm data set. It was compiled by Joanna Bennett and colleagues and then presented in this paper by Jen Sunday. And they took sort of one thermal limit per species and then looked at these large-scale latitudinal patterns across marine, intertidal, freshwater, and terrestrial groups, as well as ectotherms and endotherms, and found some interesting things. One, there's this general confirmation of what we might expect, that as you approach the poles, thermal limits decrease, right? Moving from the tropics to the poles, you have lower thermal limits. But they also pointed out that there's these really interesting differences between sort of realms, where that latitudinal drop-off in thermal limits is stronger in marine and intertidal taxa than in freshwater and terrestrial taxa. And we'll get into a little bit about that later. One of the reasons, or one of the more, one way to apply this thermal limit data is then to look at how this corresponds to vulnerability to things like extreme heat events. We know that the intensity of extreme events is increasing, as well as their frequency. And so these are sort of forward in time climate projections for heat waves. And here are the large-scale latitudinal patterns in warming tolerance, which is a metric of vulnerability. It defines how close your thermal limit is to the maximum temperature in the environment. This is a paper from Malin Pinsky and the rest of the GlobeTherm group. They found interesting things, again, that sort of towards the equator at lower latitudes, you tend to be closer to your thermal limit in marine systems, while in terrestrial systems, there's this mid-latitude sort of a anti-peak in vulnerability. Our group was really interested in what happens within species, right? If you remember, the GlobeTherm data set has one thermal limit per species. And as many of us are familiar with, there's tons of variation within species between populations. And so we started off with a relatively straightforward question. We wanted to know, are the within species patterns and thermal limits similar to the across species patterns? And so to tackle these questions, we we're going to dig into these assumptions first, and then we'll talk about what we did. So this is an important question when we're thinking about predicting vulnerability to climate change and trying to manage those impacts ahead of time rather than just responding to them. Using an across species pattern, we can assume that within species, these sort of shorter lines, that everything will line up, that everything will be parallel. Thermal limits change in a similar way within species as you see across species. But given all of these different time scales that populations have had to adapt to local climates compared to species divergences, we might also expect that divergence in thermal limits is weaker, right? So that these latitudinal changes in thermal limits are less substantial than we see across species. And then like Jen Sunday's paper pointed out, there's this variation between realms that might also be important to account for, where latitudinal patterns within species might differ between things like marine and terrestrial, intertidal and freshwater taxa. And so this is the sort of stuff that we wanted to look at. To do this, we went through and ran a meta-analysis project. We started by collecting papers from the literature that measured thermal limits in multiple populations with individuals that had been acclimated to a common environment. And so we're trying to minimize environmental effects here. There's a whole other suite of inclusion criteria if people are interested in talking about that, but these are the two main ones: multiple populations and acclimated individuals. On the right-hand side of these slides will be a general workflow for sort of what we did to help keep us on track. But you can see at the end, there's decent spatial coverage in sort of North America and Western Europe, and then some data shortages elsewhere, but some really nice exceptions of studies that did latitudinal gradient studies in different environments. We ended up with just under 1,200 thermal limits to play with, but we're going to filter those down even more because it's hard to get elevational gradients in thermal limits in marine systems, or lower thermal limit measurements are less common. We're going to exclude those studies. And so we're left with this filtered data set of upper thermal limits from studies that examine latitudinal patterns within species. And that's what we see here. We've got latitude on the x-axis and thermal limit on the y. Each study is shown with these sort of small gray lines, and the overall pattern that we saw is this colored regression in the background. And again, we start to see similar things to what the Sunday paper showed, where overall we've got this larger decrease in intertidal and marine groups than in terrestrial taxa. But again, what we're really interested in is what's happening within species. And so we're going to summarize these individual gray lines instead of looking at the overall pattern. And that's what we see here. We pulled the pattern, the parameters, the latitudinal slope estimates from the Sunday paper in these circles. And then the triangles are what we found. And there's some interesting things. One, it's qualitatively really similar to the Sunday paper, where we found stronger divergence in the intertidal and marine taxa, less divergence in the freshwater and terrestrial taxa, but with a reduced magnitude. And so the latitudinal variation thermal limits is smaller within species than it is between species. We then wanted to look at sort of what's driving these patterns. And so, for example, we have a conceptual thermal landscape here across latitude, and a distribution of populations in two different species. And we wanted to look at the driver specifically because latitude is a proxy for a lot of things, but temperature does not follow sort of uniform thermal gradients in many cases. And so, for example, this species here has a larger latitudinal gradient, but occupies a narrower thermal niche or thermal gradient than this species here with a smaller latitudinal gradient. So what we did, this is where we ran our formal meta-analysis, was looked at pair-wise comparisons of populations using a common control. So within each species, we took the highest latitude population and then compared thermal limit divergence and the difference in maximum temperatures between these populations. And so that's our effect size. There's a meta-analysis panel later today, and so I'm not going to belabor sort of meta-analysis and all that. But the takeaway is that these meta-analyses weight your points by the associated error. So it's not just the difference in thermal limits, it's going to account for variation in errors. And so here, when we looked at the divergence or the effect of maximum temperature differences on thermal limits, we saw another interesting thing, that divergence in the marine realm is explained well by differences in maximum temperature. Intertidal organisms are there is some effect, but then for marine or freshwater and terrestrial taxa, there's really no effect of maximum temperature differences on thermal limit divergence. And so that's something that's worth exploring. Other studies have shown that behavioral thermal regulation is super important for sort of the experienced environment, or for organisms that aren't capable of behavior, things like plants. Utilization of microclimate zones can really strongly affect essentially the maximum temperature that you experience. And so that might be what's driving this. But yeah, so we want to look into that more. Then to sort of finish off this across species, between species, within species comparisons, we wanted to estimate warming tolerance for our populations. And again, this is the difference between thermal limits and the maximum temperature of population experiences. But we had some methodological considerations here first. If you remember, all of our studies acclimated our animals to a common condition, and that's not necessarily reflective of what a population would experience in the field. And so we need to account for these effects of plasticity before we can estimate warming tolerance. And so we calculated an adjusted thermal limit for all of our populations using reaction norms. And Mark gave a really nice introduction to plasticity, so that saves me some time. But in general, we had two groups of studies. Some studies did acclimate individuals to multiple temperatures, and so we were able to use the reaction norms from those studies to estimate thermal limits in the field. So for example, each population had thermal limits measured after acclimation to 2 temperatures, and then we just sort of slid along the reaction norms to match thermal limits to the average temperature at each site, right, that the population came from. That gave us our adjusted thermal limits. We were also then able to model this variation in plasticity amongst populations to predict thermal limits for studies that only measured thermal limits with 1 acclimation temperature. And I'm going to come back to the plasticity model in a second. But this one had a couple more steps. Right, a study would acclimate individuals to 1 temperature. We then calculated the difference between that acclimation temperature and the mean condition in the field, and then used a predicted acclimation response ratio, or predicted amount of plasticity, to adjust our thermal limits to match those conditions. And so in the end, we had all of our measured thermal limits right under these acclimation conditions, as well as an approximate thermal limit that a population might experience in their home conditions. And so that's what we have here. We use those adjusted thermal limits to then calculate warming tolerance, right? The difference between those adjusted thermal limits, the adjusted thermal limit and the maximum temperature at a location. And we see some interesting patterns here as well, with strong differences across all 4 habitats. This latitudinal drop-off in freshwater taxa, increasing in intertidal. There's no real pattern in terrestrial, and this really distinct curvature in marine taxa. We're gonna be cautious in interpreting this across species patterns, so because we're working with a limited number of species. And so again, what we're interested in is what's happening within species, right? Do these within species patterns match what happens across species? And so we've got all of these within species slopes summarized here in these histograms. And it makes 2 points. One, there's a lot of variation within species, right? There's a pretty wide range. All of these, all 4 habitats have species that increase or decrease warming tolerance with latitude. And so that reinforces that population level thermal limit measurements are important for predicting patterns in vulnerability. But there's some other interesting differences where, in general, the median slopes, these lines here, are higher for marine and intertidal than for terrestrial and freshwater. And so this brings me back to the plasticity model. And so this was a bonus project that kind of came out of this, where we were putting it together. We were looking at the results. We're like, this is kind of cool. Let's write this up as well. And so we had a collection of studies that measured thermal limits in multiple acclimation temperatures. And this let us run a second meta-analysis on variation in plasticity itself. This is a paper that was led by Jory Barley and Morgan Kelly. And in this meta-analysis, the same kind of thing, effect sizes were estimated using both mean thermal limits and variance, so we could weight points by their by the error associated with them. And using this, they found this strong negative relationship between plasticity and thermal limits. And this is something that lots of people have mentioned already, which is exciting. But they also found that there's no relationship between plasticity and something like annual temperature range. And, so adding in something like a variance component or a predictability component of the environmental conditions might be interesting to look at to see how this compares there. And this is kind of a fun point that I've been thinking a lot about, which is as we think about responses to climate change, are increases in thermal limits actually reducing vulnerability, right? If it's coming at the expense of plasticity. So there's some cool stuff to think about there moving forward, I think. There's 4 main takeaways for our project that I want to point out. Thermal limits, warming tolerance, and plasticity all vary strongly within species. And so across species patterns can be really useful for highlighting broad scale patterns, but are maybe not an effective way of predicting vulnerability within species. And so if population conservation is a priority, we really need these population specific measurements of physiological traits like thermal limits. In the marine and intertidal taxa, this population divergence corresponds with maximum temperature differences, but there's something else going on in freshwater and terrestrial taxa that's worth exploring, and I'll point that out again later on. And then there's this negative relationship between thermal limits and plasticity that might be important to consider as we think about vulnerability in a changing climate. So the future directions, I think one of the main things that I took away, I'm biased because I measure thermal limits, but this was encouragement to keep doing exactly that, because there's some really distinct spatial gaps in population level thermal limit measurements. So the open ocean in general is completely devoid of thermal limits, and Mark mentioned stable environment, or we're thinking about predictable versus unpredictable environments. The open ocean is stable, predictable, and so might be most at risk to changes in temperature. While we're thinking about thermal limits, don't forget about variation in plasticity, because that's also super important when we're thinking about vulnerability. And then I want to give a shout out to this paper. Seema was in our working group, and this is a nice review that came out a couple of weeks ago on local adaptation, but they have a really nice section on the importance of keeping the local in local adaptation. And so going out to these sites and measuring thermal limits without incorporating insight from local communities can make those field work less efficient, and can sort of exclude really important insights into the local dynamics of those communities. And then I'll end with this quote from Niels Bohr, or someone, I couldn't find the attribution, but making predictions is hard, especially about the future. And if our goal is to be proactive rather than reactive with sort of diversity and the climate crisis, we need to think about how to make good predictions, essentially, in a really difficult system. So one of the things that we started to look at with the terrestrial and freshwater taxa is the effects of environmental heterogeneity and behavioral thermal regulation as modifying or mediating selection on thermal limits. And this is something that you can't really pull out of something like the large scale temperature data sets that we've commonly use. So the Chelsea data set is what we use for terrestrial taxa, for example. The resolution is super fine scale, but it's still not getting at sort of on top of a leaf versus underneath a leaf, if you're in the shade or in the sun. And so there's other really great packages out there to link those two scales. And so I think that's an important thing to think about. We need something similar for intertidal habitats, and then to think carefully about the scales of temperature differentiation in the ocean. And then bringing all of this together, right, making these proactive predictions about vulnerability to climate change needs to account for this environmental heterogeneity, behavioral thermal regulation, the rates of climate change in different areas, how that might interact with range shifts and ecological and population connectivity. and adding an adaptation to predictions about warming tolerance. And so lots and lots and lots of work to do on these various topics. And hopefully some of you will do that work. And so with that, I'm happy to take some questions. Again, I want to thank the RCN for bringing us together here and for giving us all the support for the last couple of years to put these projects together, as well as Hans and Ed and Lisa for giving us some useful feedback on these papers. So, some questions.
Do we have a question? You
can also go right to coffee. That's
all right. I think that everyone needs a bit of a coffee break. So thank you again for that interesting talk. So before you go to coffee, just to let you know, we will reconvene at 10:45 for our exciting panel. If you are a panelist, please get up and get your coffee first so that you can then come and set up. So if everyone can wait for people on the panel and the moderator to go get their coffee, yeah, Dan. Yes, I can. I believe you're on this panel. That's a good thing to know. This panel is sorry, I don't have it off the top of my head, challenges of integration. So if your name is Morgan, Dan, Alli, Jamie, or Stacy, please go get your coffee first. And then everyone else can go and get their coffee. Okay, welcome back. I hope everyone got a little bit of sugar energized. And so I'm bringing this over here. Somebody at the panel, Alli's going to be plugged in. Don't come this way because there's a cord here, please. So I'm like putting this here to block you all from tripping over it. Okay. Oh, cord. Watch the cord. Case in point. Okay, so watch out for the cord, please. But welcome back to the next part of the morning. I am super excited for this panel, and I hope that it stimulates a lot of conversation and discussion. And also, I hope that you all will ask lots of questions of the panelists, especially many of you are training in integrative areas, right? You're integrating ecology, evolution, systematics, mathematics, sociology, psychology, whatever it is, right? I hope that you all kind of think about how the expertise on this panel and this discussion really relates to your own work in integrative biology and integrating fields. And so ask them the hard questions, right? We are here to really have a stimulating discussion in these different topics. And so we wanted to alternate presentations with these panels to try and really facilitate lots of this discussion. So I'm really excited, but I'll stop now and turn this over to our panel moderator, Morgan.
Hi, everyone. I'll start by introducing myself. So I'm going to be moderating this panel. My name is Morgan Alli. I'm an associate professor at Louisiana State University. And I'm excited for this panel because our organizers have assembled a group of scientists who have done some really cool interdisciplinary collaborations. And so we're hoping that they can tell us both about some approaches to developing these interdisciplinary collaborations and ways that language and frameworks were important, and also maybe some challenges to communication across different disciplines. So we'll start a little bit about myself. So research in my lab focuses on physiology and evolution and ecology of marine invertebrates. We focus a lot on adaptation to global change stressors, like changes in temperature, but also changes in salinity and changes in dissolved oxygen. And we do a lot of our work on oysters and on copepods and use field experiments, also some lab physiology approaches, and then we also collect whole genome and transcriptomic data. So we're integrating a bunch of different kinds of data, but then also based on the types of questions we're asking, we also collaborate with researchers and stakeholders across a pretty wide variety of perspectives. So a couple sort of collaborations for my lab that I'd like to point out as sort of things to sort of think about ways that these could be challenges. Number one, because we work on oysters, we end up by collaborating a lot with our state-run sea grant hatchery for oysters, because oysters are just not a species you can just go and culture in a beaker or in a test tube. You really need a hatchery with a lot of expertise in producing batches of oysters. You've got to rear algae, you've got to feed the larvae for weeks before they're ready to settle. You have to actually have the sea oysters ready to outplant in mesh bags on local long lines. It's a really involved process and it takes a lot of expertise. And so we really depend on our hatchery to produce experimental animals. And we have a really big hatchery. But this is one example of how collaboration across disciplines can pose challenges as well, because the goals of the hatchery are aligned with science. They're also a science research facility, but they have a lot of other goals as well in terms of just production of oysters and production of oysters, especially for restoration. And so there's a lot of important conversations about what we're trying to get out of our research projects versus what their needs are in terms of producing oysters under the conditions that meet their needs. And then a second sort of related collaboration that I'll point out from my lab is that some of our work is specifically aimed at meeting the needs of local stakeholders. And in those cases, when we are using genetic data to answer management questions, one of the challenges we face is being really clear about what genetic data can and cannot tell you in terms of sort of conservation functions and conservation goals. So, for example, we can use gene expression data to tell us something about the causes of an unexplained mortality event in the field in oysters. We cannot, at least in my lab, use CRISPR to genetically engineer an oyster that will survive all manner of hurricane catastrophes and climate change. And so it's just been really important for us to communicate with non-geneticists managers about what we can. And so with that, I will now pass the microphone over to our first panelist, Stacy Krueger-Hadfield.
Hi, everybody. I'm Stacy. I won't repeat my whole name. I'm an assistant professor at the University of Alabama at Birmingham, and my lab is an evolutionary ecology lab. The ocean is where we like to spend a lot of our time, but as I mentioned last night, since moving to Alabama, I had no idea, being from California, that Alabama was so biodiverse. And we've lost a lot of freshwater mussels and snails and crayfish before they were even described. And the thing that we spend a lot of our time, or the types of organisms that we spend a lot of our time thinking about, are algae. Ever since I took a psychology course, that was sort of me done and dusted. And we don't really know where any of the macroalgae are. And I was talking to you, I think Jamie yesterday, about just how big of a deal that is to even begin to do any natural history, let alone do any population genetics, and you don't even know where these organisms exist. And we integrate both basic natural history observations, where we're just out looking at stuff, recording information, with population genetics and with theory. We don't really do much theory in my lab, but we collaborate with different folks. And one big project that we're in right now is developing better population genetic tools for organisms that are partially clonal. And so a lot of the organisms that have been discussed so far at this workshop are much easier to work with, in the sense that they're pretty much fully sexual, even if they're undergoing some sort of inbreeding at certain points. The problem with something that's partially clonal is that it messes up a lot of the population genetic summary statistics that we want to use. And so we're developing better tools for these types of organisms, because partially clonal covers all manner of sins, from very, very clonal to occasionally you rip yourself in half, right, if you're an anemone. And that presents a whole lot of challenges. And one thing that we always find
Okay, where did you lose sound? Just now? Did you hear gametified and sporophyte? Give me a thumbs up on Zoom. We all, Molly, I don't remember when I said we always find. So it's not. So what Molly was presenting, I'll just repeat myself earlier, is a very specific type of life cycle. And if you're an alga, you have a gametophyte is haploid and a sporophyte is diploid. And that's not the same genome. John's saying no. Can you not hear me, John? Testing. Oh, okay. Oh, hey, they confuse everybody. It's not the same genome, and that presents a whole host of challenges because for a lot of these organisms, you really need to be an expert in distinguishing the different stages and distinguishing the different species. And so you have to integrate some taxonomy with basic natural history, with population genetics. And then you add in the complications of your typical population genetic tools. And the way that pretty much everyone here has been discussing dispersal is not how we think about dispersal in macroalgae. And so it presents this whole other set of jargon that if you speak to somebody who works on angiosperms, they have all this information because people were crossing plants in the mid 1800s. And then they use genetic tools. For macroalgae, we started working on them, I mean, before population genetic tools were common, but we don't have that repository of information of who's mating with whom and even what stages are present where and why some stages are present here and not here. And that really complicates any population genetic interpretations. And then you have the added complexity of trying to integrate botanical and zoological terms. And then there's a whole host of phycological terms that are layered on top of the botanical terms. So then speaking to somebody who works on animals is always a challenge, and how people use terms, which is where, if you read the paper that Molly and Letitia led, that first box came from. And I've heard a couple of people here say life histories, life cycles, as though they're synonyms for one another, but they're not really. And then figuring out a way to use those terms so that we can start to understand a little bit more why there's so much diversity across the eukaryotic tree, which I was alluding to yesterday evening, which I think requires being very explicit about what a life cycle is, and then figuring out how organisms go around that life cycle, which is really just a synonym for talking about sexual reproduction, because it's meiosis and fertilization that lead to that cycling between a larger and a smaller ploidy stage. And how you do that is tremendously variable. And even though we have good theoretical predictions for why that happens, we don't actually know what happens in nature. And so sex in nature is still pretty much a black box. And I think that's largely because of the types of organisms that we work on. And so I'm happy to talk more about how to use terminology effectively and precisely, and then how to work in a system that we're missing a lot of the basic information that even we have for larval dispersal. We just don't have for some macroalgae. So.
Hi, I'm Jamie Pringle. I'm an oceanographer at UNH. Trained as a physical oceanographer, though starting with my postdoc, I have spent about half my time doing interdisciplinary physical biological coupling. And so I've spent a lot of time wrestling with terminology and trying to figure out what other folks mean and trying to write so that other folks understand me. And a lot of that is just simple vocabulary. What dispersal means to a physical oceanographer is very different from what dispersal means to a biologist. For a physicist, dispersal is one of many processes that move things, and it's separate from advection. And then, you talk to a population geneticist, and there's Tajima's Greek letter, and all of that is difficult, and you need to be mindful of it when you're writing, but is fundamentally straightforward. They're words you need to figure out how they're used in different communities. You need to figure out when different communities think they're using the words in the same way, but they're really not. And so you need co-authors who are in those communities and oh, this doesn't make sense, or what are you saying? But again, all of that, if you care and you listen, you can deal with. The more fundamental problem is when people have different ideas of the origins of their terms. And so one I'll focus on, and this happens a lot in genetics, but one I'll focus on right now is a population size. So often in genetics, you're looking for an effective population size, which is based in a model. What is the effective population that makes my model predict the right amount of diversity? Or you have a simple model and you know it's a simple model of a population that is panmictically interbreeding. And you say, well, the number of genes will have something to do with N sub E and a mutation rate and all that. That N sub E is a model-derived thing. If you go and talk to a physical oceanographer and you say, what is the size of the population in the retention region which provides the diversity? They're going to at least conceptually think about, well, it's this bay and 1 little barnacle, 2 little barnacle, 3 little barnacle. And that's a very different quantity. And bridging that gap from parameters that we choose, but whose ultimate origin is a model. The model predicts something we observe, and from that we infer a parameter. So you observe diversity, you assume a mutation rate, you estimate an N. That's a very different thing. Now it's somehow related to critters on rocks. But when you talk to a physicist, they're going to start with the critters on rocks. And so that bridge between things we want to know that are in theory observable and things we want to know, which are parameters in a model. And I'm not saying any one is better or worse, but there are different things. And that's a gap we need to break. And often the reason the gap exists is it's really hard to understand what actually sets an N sub E in the real world, because everything affects it. All sorts of aspects of history, variability from generation to generation. It's hard, but that's the gap we need to start to bridge if we want to start to attach the genetic observations to real world dispersal dynamics, or at least have those conversations to see how far we can get.
All right. I'm Dan Bolnick. And my group at the University of Connecticut is an evolutionary immunology, evolutionary ecology group. And there's a couple of perspectives that I want to take on here. One is we have a project that's been going on since the start of this year, funded by the National Science Foundation's Rules of Life program. And the Rules of Life program explicitly requires that your proposal be collaborative with PIs drawn from people who would normally go to different divisions of NSF. So I couldn't have 2 people who normally get funded by DEB. I couldn't have 2 people, one DEB, like evolution ecology panel, and another who normally goes to integrative organismal systems. We're talking like not biologists. And I'd thought about these Rules of Life grants for a few years and never really found a natural, it felt very forced to me to create a collaboration out of thin air. And I don't know what a physicist does really. And so luckily, I started talking with some people in network science facilitated by Sam Scarpino, who was a former student with me. And he connected me up with some network science people. And so we ended up putting in a Rules of Life grant with 3 PIs, myself, and then Tina Aliassi-Rod at Northeastern University, who's a computer scientist specializing in machine learning of network architectures. And then a statistician at the University of Wisconsin, Yaoyan Wang, she specializes in developing the mathematical theory for exponential random graph models of networks. And so how do you actually do a collaboration? A, we're not at the same institutions. B, we don't speak each other's languages. And I have to say that, like, as an evolutionary biologist, evolutionary geneticist, I just kind of got in the habit of using words that all of you probably are familiar with. But when I start talking to a network scientist and she says, hold on, RNA, what's that? And I went, oh, and she hadn't had a biology class since freshman year in high school. So I had to back up and say, okay, let's talk about what gene expression is. And she says, okay, well, explain genes to me again. So this is a really grounding moment of like, okay, but it's not just that she's ignorant about biology. As much as I've read a couple of textbooks on network stats, it's a pretty big field. It's very fast moving. It's fast moving in the sense of like, if you're not publishing a new paper every month, you're not keeping up. And so the process that we engaged in was a really fun one, which is we started a reading group, and this reading group is on Zoom because we're in different states. And every Friday afternoon, we have a reading group. The first semester, we read a pair of textbooks on network statistics. And then we've been doing a paper a week or 2 papers a week. And sometimes they are network stats papers from the Journal of Complex Networks or the Journal of Statistics or a computer science journal. And in computer science, often you just publish like a 2-page meeting abstract, and that's your publication. And that's enough. And that just comes with a bit of code and you're done. So the publication styles are very different. So we're reading very different kinds of papers. And every week we have about sometimes 5, sometimes 10, sometimes 15 people. We discuss this. And usually it's built as like, okay, today is the biologist's day to teach something to the computer scientists and statisticians. And today is the statisticians' week to teach something to the biologists. And it's been a really fun process. And I have to say, if anyone is really into network computational issues, this is an open discussion group. It's not just the 3 labs that are involved. We've gotten people from California and people from Europe involved. So let me know if you want to talk networks. We have this open reading group on Fridays. It's a lot of fun. Let me know. I was actually earlier today working on the email saying, hey, summer's over. It's time to restart this. So it was really grounding to realize the extent to which we just need to teach each other our basic vocabulary. And we need to teach each other the differences in publication expectations. A good journal for my lab to publish in is not the same as a good journal for Tina's lab to publish in for their career advancement. And in computer science, literally, they're just firing off these short little papers one after another. Whereas we have to say, look, we need to spend 3 years collecting samples, and then we need to do the lab work, and then we send it to the sequencer. And they keep saying, well, where's the data? Where's the data? And I'm like, well, we're working on it, but I'll have it to you in a year. And to them, that's forever. So the cultures are extraordinarily different in terms of how you get credit towards career advancement even. And you need to negotiate that with people and have clear understandings and expectations. And you don't even know those differences in culture until you sit down and start working together. So it's been a lot of fun.
Hi guys, I'm Alli Cramer. I am a postdoc at Friday Harbor. And you guys have done a really good job kind of summarizing, I think, a lot of the points we're going to talk about in the panel. So I'm not going to repeat stuff that they've mentioned, but I agree with all of it. But my specialty is in ecoinformatics, which is essentially, you guys are probably familiar with bioinformatics, which is very focused on kind of genetic type biology data. Ecoinformatics, we use all sorts of data sets, and we combine them and use data science and data synthesis to gain new inferences. And it's really focused on kind of leveraging all those large scale data sets that we have all around the world from these kind of large observatory systems. But the problem is a lot of these data sets don't actually answer complete questions, right? They are one component of a question. So you need to work interdisciplinarily to actually ask interesting integrative ecology questions, which is kind of where I sit. So you already heard my colleague Jennifer Hoey discuss the work we did, combining the kind of genetic and population gen perspective on connectivity with the movement ecology perspective. I've also done work on the lakes. So there's a data set that I'm one of the lead team members on, which is a global lake climate population data set. It's 1.4 million lakes. And we combined a data set on lake surface area with satellite derived data sets on temperature, precipitation, and socially derived data sets on population size, and created this enormous data set, which we published. And that was multiple people from different fields. And I've also worked on a lot of stuff with MPAs and a lot of, marine protected areas, and a lot of kind of integrating research from ecology and sociology perspectives, where, because my expertise is data synthesis, kind of act as the bridge. It's a lot of computer science, but ultimately it's basically classification, which is how we view these terms. What does this term mean? What is the context the term comes from? And what is the utility of the term? We don't always have to make a brand new, what we call a data dictionary or a brand new, definition glossary. It's more important that the people in the collaboration understand what they're talking about. And then when you pick a target journal, or you pick, an end goal product, you use the language of your target audience. So it's very much about communication. And I think that, that aspect of kind of integrating across research requires not just communication, hey, can we all agree on definitions, but like Dan was referencing, communication, hey, what are all of our goals? And what's the ultimate outcome that we want from our project? And with that in mind, you can normally come up to some sort of consensus on what the actual language is that you're going to use as you move forward. Yeah, I think that's all I'll say for the moment, but good job.
So I have a couple questions for our panel, but let's throw it out to the audience or over Zoom to see if anyone has any questions you want to ask a specific panelist or put out to the whole panel.
Hi, this is Diana, and I have a question for Dan. So I'm impressed by the amount of effort you're putting in to learning about network systems. And so I guess one question would be, you obviously have to be very strategic about doing that for every potential collaboration that would be interdisciplinary. So how do you, choose where you're going to put that effort, so that there might be some collaborations where it's a little bit more cursory, maybe if the language is a little bit closer, and then others where, you do go through this whole process of really learning how to collaborate with that group.
Great question. I'd say a second grant that we have from NSF through Ecology and Evolution of Infectious Disease is also collaborative. It involves somebody from veterinary medicine. It involves a mathematical ecologist. We all speak pretty similar languages, so we're not doing that. So yeah, you hit the nail on the head. Part of it is, well, network computer science and abstract stats are just far enough out there that I have to learn that stuff if I'm going to be effective at communicating. The other part is, honestly, we're all here because we like learning stuff, right? And so strategically, I spent a lot of my career hopping to new disciplines and then hopping to another discipline. So at some point in my career, I thought I want to learn immunology. And I took a couple of years and I just read immunology textbooks, and I went to immunology conferences instead of to evolution and ecology conferences. And this is maybe that next step in that evolution. It's, you pick a thing that you want to learn next, and then you have to invest the time in it. And you can only do that for really one thing effectively at a time. Absolutely.
I have a very general and maybe very silly question. I mean, you all talk very wonderfully about the challenges which you have once you already have a collaborative project in mind. But how do you come up with the original idea of the collaborative project? How these things are born? That's what I want to know.
Randomly and with an open mind. This is Jamie. So one set of collaborations on interdisciplinary stuff happened because Peter Franks at Scripps had money for a postdoc. Another occurred because my sister knew this guy, John Wares, who had some questions about the ocean. And others have occurred by sitting around in meetings like this and drinking beer afterwards. And others have occurred because I've been reading something in biology where they've said something about what the ocean was doing, like, you know, things that drift in the water off of Point Conception for 2 weeks move on average 50 meters. I was like, but no. So randomly and by talking to people with an open mind, and just things will come your way if you pay attention. The harder question is actually, how do you conserve your finite bandwidth?
I think randomly a lot of connections people you run into is really important, but I also think it really helps to, like for my particular work, I like to talk to people who know nothing about anything that I know about, because the questions they ask tend to be fundamental and tend to be ones that we have very strong assumptions about. And then when you actually go look, some of those assumptions are supported and some of those assumptions don't have any actual data supporting them. We just kind of moved on and assumed that that was the case, right? So for the lake population data set or climate population data set I mentioned, there's a couple, that's a derived data set. So there were pre-existing data sets and we connected all of them with this giant brute force computer work. That came about because someone in a lab meeting who studies agriculture runoff, so they're not limnologists, they don't study lakes, made an offhand comment where they said, well, we all know that lakes waters around the world are decreasing dramatically. And then the limnologists in the room were like, well, do we actually know that? Wait a minute. Well, how would we find that out? Turns out they're wrong. On average, lakes are actually increasing, but certain places are dramatically decreasing. But those kind of things where you're talking to people outside of your discipline, dramatically outside of your discipline, can really help you think of integrative questions, because those fundamental assumptions we make are right at that interface of multiple points of view.
To add to that, going to conferences is huge, and in particular, push your boundaries. So some of the most valuable conferences I've been to are conferences that are not in my area. If I go to ecology and evolution meeting, yeah, I get collaborations out of that, but they're not interdisciplinary in a profound way. The really interdisciplinary ones are when I go to a immunology conference, or I go to a Packard Foundation meeting that has computer scientists and physicists and astronomers and geologists all in the same room for a few days. And then you meet people who are wildly different. And of course, almost all of those network connections that you build, they're just kind of casual chatting, but every now and then you just strike gold. The other thing is, honestly, some of my collaborations are word of mouth, friend of a friend, colleague of a colleague, or I have gone online and thought, okay, I need a plankton disease ecologist. Who's out there who does that? Okay, here are people who I know. Here's some people I kind of know. And then I've cold emailed people. Miaoyan Wang at Wisconsin, I just figured out who's doing the best pure math of network statistics out there.
So some of us have experienced great collaborators, and many of us have experienced those not so great collaborators where we're like, all right, that was a one-time experience. How do you vet potential collaborators that are not in your field? Because right now, when you're a potential, let's say you're a graduate student or postdoc thinking about collaborating, you ask your PI like, oh, what's this person like? How are they? And you kind of feel it out. But if you're talking about across disciplines, how do you vet potential collaborators, Jamie?
You have to be prepared. Well, you can talk to people. You probably know someone in their university or somewhere close, especially if you are at a university where there are big departments. But, some collaborations don't work. And that's and then you move on. And the issues why they don't work are often idiosyncratic to you or idiosyncratic to the professional or life stage the other person is at. Not everybody who you collaborate with can collaborate with you right now. The other thing I would say is I wholeheartedly endorse Dan's idea of pushing yourself out. I tend to alternate between doing that and doing something core in my discipline. I just find I need to keep that side of me up. So I sort of flip-flop between strategies over time. But yeah, figuring out who to collaborate with, it's like dating. Sometimes it works, sometimes it doesn't.
Not to step back a minute, but I was going to add to what Dan was saying, the serendipity of meeting people and cold emailing people, and the population genetic consortium that I'm part of was a connection from where I did my PhD, but I've done work now with this one theoretical population geneticist, and we don't speak the same language at all. I mean, he speaks French, so that complicates it. But even when we're speaking the same language, if it's French or English, we're still not speaking about the same thing. And so it's really caused me to have to step back and think how I'm defining things, how I'm explaining things. And it's been uncomfortable, but I think that has made it much easier to communicate things that I find most people don't understand when I'm talking to them about it. And I wasn't entirely sure all the time. Is that because I'm not explaining it clearly and it's really this self-evident thing that I'm making more complicated? Or is it something that is actually confusing? And I think it was probably both. And over time, you get better at explaining things. But Molly had a question, which I just wanted to make sure we had sort of answered. Can you give us a thumbs up? Okay. Do you want to ask the next question?
Yeah, sure. I think we've got one more question from Jimmy DeBeo in the chat. Does anyone else in the audience before I? Okay, I'm going to read Jimmy's question. Jimmy says, it occurs to me that people within a discipline can disagree about language or fundamental terms for certain principles. What is the best way for communicating that to those who are outside of a specific discipline, or choose a definition to communicate?
For you. So this is something that we encountered with a paper that Molly talked about earlier. And part of the reason when I heard Adam Reitzel at the last meeting talking about life cycles and complex life cycles, when you read Bob Podolsky's paper on this, the other papers that Molly was alluding to, it's always metamorphosis and it's always an animal. And that's not the only way to be complex. And so we had arguments, and they were like friendly, good arguments, but they were still arguments about what is a complex life cycle, what is a life cycle versus what is life history, which is where that box originated, because I was very, very adamant that we clearly defined what we meant by a term so that it was clear to somebody reading that paper. And then hopefully that paper would be read for more than just the types of experimental designs that you could use, but also to be much more intentional with how we're defining terms. And we didn't all agree. I think we came to a consensus in the end, but Molly can attest to this as well, that when we had our Zoom meetings about this paper, we went back and forth about just that box. And even some of the reviewers on it thought the box was like not necessary because a life cycle equals a life history equals a life cycle. And everybody knows this. And David Haig wrote a really good paper. I think it is from 1997. I'd have to go back and look it up, but about how like common knowledge is often common misconception, and then went through all these examples of where we all know something, just like what Alli was saying, lakes are all decreasing. And we make these maybe true in this specific instance, but not otherwise. And the thing that I've tried to do based largely on the working group that we had from the last time we were out here is to specifically define a term. And we've had some reviewers and papers say that's not necessary. Everybody knows what this means. But it's clear that they didn't because they misinterpreted what we meant by the term. And so I think just defining that and then coming to a consensus within your group and having a clear, precise definition, then anybody who's reading that paper or is outside of that consortium might understand.
So I want to just be said about defining your terms, and any reviewer who has a complaint like that is wrong. And I break the question up into two parts. One, how do you deal with disagreements about what a term means within your field, however you define it? Those are ones. And that's troublesome, and reviewer number 2 will be an ass about it. And that's just very difficult. So I'll skip to the easy one. For between field issues, which happen all the time in interdisciplinary work. So what a physicist means about dispersion is totally different from what a ecologist means by dispersion. And you just have to accept that and let it go. Write for the audience of your paper, but most importantly, define it. So I just did a paper with a bunch of folks looking at transport across the near shore. It's an annual review paper. And we use dispersal in the physics way, the random stochastic movement of particles. And we define it very carefully because it's for an interdisciplinary audience. And we're saying, this is our dispersal, and this is what I mean. If I'm publishing in a biological journal, I will use the appropriate ecological definition of dispersal, because don't be an asshole, right? Go to your audience, meet your audience, but be clear about what you're doing, so that when your audience isn't who you think it is, it's some random physicist trying to get into the game, they will see dispersal and they will know what it means. So you should be aware where those breaks occur, where they are, meet your normal audience and define it carefully enough so that everybody understands what you're talking about.
Sorry. Go ahead. So I'll tackle the difficult one that Jamie just alluded to. So in my experience, when people disagree within the same discipline, it is the harder one. They have very impassioned defense of their point of view. And so that's where it helps to not be in that discipline, number one, because you can ask lots of intentionally ignorant questions. Maybe you can go something you actually know. So if you're trying to like function as a moderator in this situation, there's a paper I was on, the marine protected area paper that we referenced. And that paper involved socio-ecological networks in this really unique area of the Baja Peninsula in Mexico. And there are these really cool marine reserves. That's what they're called in Spanish. They are marine reserves, and they rotate. They're also rotating fisheries closures. If you work in what Americans call marine reserves, which are very specific and never get opened up to fishing, and you work in fishing, where, yeah, we closed it for a couple of years, the two people on that paper had extreme feelings about whether or not we should use that word. That is the word it was in Spanish, but we ended up at an impasse. And the way you're able to kind of address that, I think, is by zooming out and just say, okay, you know what, we're not going to use either of those terms. You guys are both right. Those terms are bad terms. When we zoom out, what is the commonality? And if you come at it from a perspective of, gosh, I just don't know what you're talking about, I'm so confused what the difference is, you can actually get your collaborators to agree as they teach you what this commonality is. In this case, it was no-take zones. So that's the word we ended up using. A no-take zone covers both of these scenarios. And that only happens because you're forcing those people to realize what they have in common by communicating it to the other people who are not part of the discipline. So I think that trick as a moderator, as someone who's doing cross-disciplinary collaboration, is really valuable. Now, if it's in your discipline and you're attached to it, maybe it's a harder role to have, but it's a really good way to get people to agree, because there often is something on one level higher that people do have this kind of core commonality about.
Real quickly, just to inject some humor here, I imagine most of you have seen the movie The Princess Bride. You know, the scene where he keeps saying inconceivable, and the guy's like, I don't think that word means what you think it means. So that's a really deep issue and very widespread. And I'm going to put in the chat window here a link to a paper by Yoel Stewart in the American Naturalist, who looked at the word parallel evolution. And we all think we know what that means, but lots of people get confused by it. He looked at the use of that term across 150 years in the American Naturalist and found that at any given point in time, there are like 5 distinct uses of the word and that they changed over time. And it's just a good lesson in, for almost any word that you think is obvious, there are going to be people who think it's obvious in some different way. So define your terms. Absolutely. As editor, and by the way, I'm editor of the American Naturalist. Come talk to me if you have a paper you want to talk about. As editor, I'd say this is one of the #1 problems that people run into. And the editorial solution is always define it. This is how I'm going to use this word.
And just one last thing. If you're in one of those heated discussions about your term, ask yourself, is this really the hill I want to die on? Is anybody learning anything?
More questions from the audience?
I was just thinking about when I was a grad student, I wasn't always good at communicating what didn't make sense to me because I thought it didn't make sense because I didn't like study enough or I didn't read enough or I didn't know something. But from what you're saying, it's like very important to communicate and be adamant about what doesn't make sense to you when you're collaborating with someone who's has different terminology. So could you just speak a little more to the importance of like how to communicate that maybe for the younger people in the audience?
Sure. So I think I've avoided that imposter syndrome by never working with people who were in my discipline. No, not quite, but yeah, it helps a lot when you're the only person who represents your discipline. But that's not really a trick we can do all the time. I think it's important to recognize, #1, that as we've demonstrated in this panel, that people don't actually agree and don't really know what they're talking about. Like they don't fundamentally, some of the core terms, we talk past each other all the time. So if you're a grad student or an early career researcher and you're trying to, maybe the question's because you're trying to guide the group, maybe the question's just because you didn't know what it meant. Remember that there might be other people, but that knowing that isn't necessarily always helpful, right? Reminding yourself of that when that little voice is in your head telling you there's no one else here. And so I try to frame the question. I try to guide the question instead of being more focused on me, like, hey, I'm having a hard time doing this, unless I'm using it as some sort of like moderation technique. But in general, if I don't know, I'll be like, instead of me not understanding this, I'll be like, hey, are we clear on what this term means? And I think making the group, putting the onus on the group to be clear about the term can put it off of you, but help you actually address what it means. And you'll probably find that it's really useful exercise. The other thing is if you're guiding a collaboration or if you're beginning a collaboration with other graduate students, have that as part of the things you guys do first. Make sure you guys are all on the same page at the very beginning. It's a useful exercise and it's also useful for, maybe you do need a glossary and look, now you've got table 1. Like that kind of exercise is not wasted time. And that kind of begins the uniting of terms without putting the onus on any individual person to not know a particular thing.
I'll just say, when you have an argument about what a term means, you should think if it's a definitional argument or it's a worldview argument. Very often what you're seeing is people who have different models in their head, even if they're null models, different models in their head of what the world is doing. And so often when you get an argument about nomenclature, it's really a difference in an underlying idea of what's going on. And so it's worth poking a little bit to see what's going on.
I was just going to add to what Katie was asking. I think it's a skill to be able to speak up in a room. And when you see other people who are speaking up in a room, they're either just more comfortable from the get-go or they've practiced it a lot. It's just like reading a scientific paper. The first time you read a scientific paper as an undergrad, it's this daunting task that takes you multiple days to really understand all the components. But then as you do it more and more, it gets easier. And so I think speaking up, there's always somebody else in the room who doesn't understand either, but is maybe a little bit more shy or a little bit more inexperienced. And I thought you maybe thought I didn't know.
No. I was agreeing.
And I think that that's just like skill building to be able to speak up in a room. And I did my PhD, for those of you who don't know, in France. And so I was always on the back foot. So I was always asking questions because for the first year I was there, I had no idea what was going on outside of the lab, in the lab, because the lab did not speak English. And so I was always confused. And so that I think grew me up really fast. But I think also find a group. So one thing we've started doing in my lab is reading about, we think about the evolution of sex. And so we've been reading textbooks on sexual reproduction and basically how everybody talks about it. Everybody's talking on different levels and about different things and just talking past one another, like ships passing in the night all the time. And I think that's been really helpful for me and everyone else in the lab to have these conversations, because then they've realized that there's a whole book about this where the first chapter defines terms because most people get it wrong. Like a term that you think, oh, I know what meiosis is. And if you think about sexual reproduction, well, it's to generate diversity. But it doesn't always. And so we have all of these ideas, and just talking about those hopefully also facilitates younger people or younger scientists to feel more comfortable asking questions. And so I would recommend that you get in a group that is also friendly. And if you find somebody who makes you feel like you're an imposter, then maybe try not to work with that person, like exit that relationship, because you want to be around people who question you and challenge you. But challenging you is not making you feel dumb. And those are two very—
Hopefully you all have chosen a question for your table. I am just going to go ahead and call on tables. Hopefully we'll have time to get to each table. I'm going to start with this group right here.
I haven't quite formed a question in my head yet, but something that this discussion is maybe think about is in my lab meetings at UC Davis, we had a social scientist come to our lab, and she was interested in studying how scientists study ecotoxicology. She was interested in that process and how we go about defining terms, especially. And at the end of it, she wrote a paper talking about like how the use of some words in science can be misinterpreted by other fields completely out of science. So one of the terms that came up was the word feminizing. So there are chemicals out there that people have shown has caused male frogs to produce more estrogen, resulting in differences in their behavior. And so in the scientific paper, they use the word frogs becoming feminized, and then social media sort of latched onto that, and all of the social media said, oh, this chemical is turning the frogs gay, which is really bad. That's obviously not what the scientists were trying to say, but sort of recognizing that the terms we use in science should maybe, we should just be more careful with the terms we're using and how they can be interpreted in different ways outside of science. So yeah, I don't really have a question, but yeah, comment.
So that's an endemic problem. And let's start with the most basic one. What does significance mean in a statistical test? I think statisticians should just suck it up and choose a different word, because in English, significant means important, and in statistics, it does not. It's a horrible problem, and we're stuck with it. And the majority of reporters you talk to, especially if they're not science reporters, are often motivated by things other than just clearly describing your science. So you just have to be careful. That's going to be forever. I hate the way biology comes up with complicated, indecipherable Latinate phrases, but it does have the advantage of not being loaded with other meanings. And perhaps if you had to start it all over again, you wouldn't choose feminizing. You would translate it into Greek, so you wouldn't have that issue. But I don't know. That's sort of security through obscurity. It's a hard question.
I mean, I agree. I think in some cases, the onus is on us to change the terms. And I think the good news is that terms do change. One of the classification papers I worked on, which is based on the work of Whitaker, he used the term physiognomy all the time, which means like the physical body structure of things and the way they're shaped. That term is really tied up with phrenology, which is the shape of skulls and horrible racist dividing of different people. For reasons that were not social, it kind of fell out of fashion. And then when it came back, people decided not to use physiognomy anymore. And they use things like body plan or bow plan or morphological features. So the good news is that they do change. But I agree, it's something we need to be careful of. And I think if you have the opportunity to try to change it, especially if it's, I mean, let's be honest, it's not just talking to reporters. It can get a one good sentence can get picked up on social media and completely misused. So read your stuff with an with an eye for pithy sentences, and they might be really fun in our field, but remember, it might be a problem. Yeah.
So pertinent to the topic of this meeting, a lot of you study local adaptation, and it's easy to start talking about selection against immigrants. That is what's happening. It's just some people want to see that as applicable to humans in ways that one doesn't want to go down that path. The terminology of eugenics is pervasive in evolutionary biology. If you take a molecular evolution class, you will read about positive selection and negative selection. That has its roots in eugenics writing. Discriminant function analysis literally was to discriminate human categories. Linear regression analysis was from Francis Galton, talking about regression to the mean in human characteristics. Like, we have a body of words that are derived from a past that is not perfect. And so it's just a matter of clearly defining your words and being cognizant of the ways that they might be misinterpreted and try and head it off, especially on social media with journalists.
I'm going to choose this table next.
So this might not be the most well-formulated question yet, but the three of us are PhD students. And so we were hoping that you might have maybe some targeted advice to PhD students or early career researchers who are perhaps not as established in their fields, or maybe don't know as many people both within and outside of their fields, and thinking about interdisciplinary collaborations. And so we were thinking, like, how would you advise your students when thinking about splitting their time, acknowledging the other things we have going on with our dissertations, in both like finding those collaborations, expending time in them, and also if you had any remarks on maybe your earliest collaborations that were interdisciplinary. That might have been a lot, but
Your postdoc not being in a field as close, but exactly the same as your PhD. And then when you're talking about your PhD work, presented in interdisciplinary audiences, in a way that is clear to the other disciplines. And that is hard when you've been so focused on your one thing. And hopefully you have an advisor or colleagues to help you do that. And then talk to people at meeting about what they do, because everybody likes to think their work is interesting, but also about what you do. Blatant self-promotion is a useful skill, and it doesn't come easy to some people, and life is not always easy. So schmooze and present your stuff to others in a way that is outside of just your narrow field. And that's a useful skill for almost whatever you do.
I would add to that. I think comics call it like a tight 10, like stand-up comedians, that they have this like tight set that they get up and they do every time. I would really work on your elevator pitch. And I think this is really important for students who've been students during COVID, because you haven't been going to meetings and you haven't been doing things that were quote unquote normal 3 years ago. And that was one thing that I discovered with 2 of my students. We were just in the field, and I didn't think I had them practice. I teach a science communication class, and they practiced it in the class, but then their projects are still new and they were a little bit uncertain, and their elevator speech was like not a tight 3 minutes. It just kind of rambled. So I would work on having, a very short and sweet thing that you can say to somebody who's not even a biologist, and then somebody who's in your very specific field, because you want to be able to use jargon to show that you can speak that language. And so if one of my students goes to a phycological meeting, they shouldn't call a sporophyte and a red alga a sporophyte. They should use the right term. But then if they're talking to somebody who knows nothing about biology, they need to come up with something even other than sporophyte, because somebody who isn't a biologist isn't going to know what a sporophyte is. And you don't need to go over the derivation of like where that word comes from.
So the old Confucian saying, a journey of 1,000 miles begins with a single step. I think it's very hard to like launch straight into, I'm going to be an interdisciplinary scientist who's mastered 4 different disciplines. No, pick one, do your PhD in it, get really good in it, maybe 2 that you're at the intersection of, but I don't think that it's always in a position to be really interdisciplinary as a PhD student. That's a phase of your life where you become the world expert in a thing. And then you have a foundation on which to build bridges to other people who have different foundations as yourself. But don't think that you need to do all of this all at once. It's a lifetime, it's a marathon, whatever analogy you want to get. Just think of it as this layer, and you're going to scaffold on new things as you develop in your career. And it's okay to say, I'm going to do this thing really well right now, and then I'll worry about interdisciplinary collaboration. And a lot of collaborations require big grants. You got to pay people. You got to pay people from other disciplines. That's something that happens at a later stage in your career.
So I'm at a slightly earlier stage than these guys. So I'm a postdoc, but I think it's important to know a couple of things. Number one, I did a master's first. So I did a master's that took 4 years. Then I did a PhD that took 5 years. I've already done 1 postdoc and I'm on my 2nd postdoc. So first of all, that's more time than probably a lot of you grad students. So these things take time to make connections. Also, like Dan was saying, don't just jump in to collaborate with everybody. Also, don't collaborate with people who are immediately in a completely different field than you. There can be really useful, really challenging collaborations with people who are just one field over, right? And so the people who are around you, like the lake project I was on, I happened to be located in the engineering building because it had the really fast internet connection to the high performance computer. So I was the lone ecologist in the engineering building with a couple climate people and some limnologists. That's how that collaboration came about. So the people who are around you in grad school can be really useful to begin kind of flexing your collaboration skills. Also, you can look for opportunities that are supported by your mentors for training. NSF has a number of them. The sociology paper I was mentioning, and in addition to the paper that I put on the screen yesterday, both of those papers came out of what's called Eco Days, which is a collaboration where they get a whole bunch of ecologists together and lock us in a room in a basement in Hawaii for a week. And a whole bunch of collaborations come out of that. And those are all technically ecologists, but there are people who did stream ecology and bacterial ecology and, disease ecology. And even those differences between disciplines can be really fruitful and make new connections, right? So look for training opportunities and start small and start with people around you who do something just that little bit different.
And you're also not going to do every single part of a big integrative group. And that's something that was hard for me. When you're a PhD student, you do all parts usually your project, and then you transition to being a postdoc. And then if you're working the population geneticist I was talking about earlier, I can't do the complex math that he does, nor do I have interest in it. And so it's okay to be the person offering biological insight into that, or into what his math is, so that it makes sense that what he's actually deriving is testable when you go out into nature. And so you don't have to do every single part. You should be conversant in what somebody's doing. And maybe Dan could speak more to that in computer science. You need to know what they're doing, but you're not going to do all of those parts. And that's where a collaboration is